Let's be honest, when you think of a creature created in a secret lab, what comes to your mind? Frankenstein's monster, the fly, or maybe Dr. Manhattan? But the truth is, in the real world, the creations that scientists mess around with are often far less dramatic. Some are surprisingly small, like modified bugs, while others look cute, like transparent frogs. But the deeper you dive into the world of lab-grown life and DNA tampering, the more unsettling it all starts to feel. The idea of building life or tweaking its code isn't just futuristic, it's loaded with consequences. So, let's take a step into that strange space where science meets something darker. From glowing pups to one infamous cloned sheep, here are 15 terrifying monsters created in secret labs. Number 15. The Soviet two-headed dog. In the 1950s, Soviet surgeon Vladimir Demikov conducted a series of grotesque experiments involving dogs, and one in particular shocked the world. He surgically attached the head and front limbs of a puppy onto the body of a larger dog, creating what the media dubbed the two-headed dog. It wasn't a puppet show. Both heads could move. Both drank water. For a brief time, both lived. The experiment was a brutal attempt to pioneer organ transplantation. Demikov believed that by pushing the limits of surgical endurance, he could learn how to save human lives. What he created instead looked like something pulled from a fever dream. Over 20 surgeries were performed, many ending in failure. Even the most successful dogs survived only days. Animal rights didn't exist as a concept in that era, especially behind the Iron Curtain, and Demikov's work was defended as progress. But the image of a living, two-headed animal shocked global audiences and haunted the pages of medical history for decades to come. Number 14. Fluorescent puppy. In a South Korean lab, scientists created a dog that could glow under ultraviolet light. Not metaphorically. By inserting a fluorescent gene from sea anemones into dog DNA during the cloning process, they produced the world's first transgenic dog. The goal wasn't a novelty, it was biomedical research. They wanted to see whether foreign genes could be successfully expressed in mammalian bodies, potentially opening the door for modeling human diseases in animals. The result was a puppy that looked normal by day, but lit up under UV light. It confirmed genetic expression, but not without controversy. These weren't test tubes. They were living animals. Cloning alone carries high failure rates and physical abnormalities, and introducing extra genes adds more risks. The fluorescent gene itself is harmless, but the ethical questions remain sharp. Is the ability to make a glowing dog a sign of scientific progress, or a line we shouldn't have crossed in the first place? When the body becomes a testing ground, the soul tends to be left behind. Number 13. The glow-in-the-dark monkeys. You can take the idea of what they are from just their names. A team of Japanese researchers created monkeys with jellyfish genes inserted into their DNA, genes that made them glow under blue light. But the real breakthrough wasn't the glow, it was an inheritance. These monkeys passed the trait to their offspring. It proved that not only could genetic modifications be inserted into primates, they could be stabilized across generations. That opened a gate to future possibilities most people weren't ready to consider. Genetic conditions could be studied more precisely and modeled more consistently. But it also meant future animals, and theoretically, humans could be modified for life. The glowing was harmless, just a visual marker, but it was also a warning flare. These weren't just individual test subjects. They were part of a long-term genetic legacy that no longer belonged solely to nature. The monkeys didn't choose to glow. They didn't choose to be born with an altered code. And yet, their existence became proof that humanity's handprint can now extend past birth, etched directly into the blueprint of future lives. Number 12. Genetically modified dogs. In a research facility in southern China, scientists altered the DNA of beagle puppies, not to treat disease but to enhance their physical traits. They used gene editing to remove a gene known as myostatin, which normally limits muscle growth. The result was a set of beagles born with exaggerated musculature, bulky limbs, powerful jaws, and a build unlike anything seen in natural dogs. The goal, according to researchers, was to develop stronger working dogs ideal for military, police, or search and rescue use. But the question looming behind this muscle-bound experiment isn't about utility, it's about intent. Dogs have already been bred for generations to suit human needs. This marked the moment when selective breeding was replaced by code-level manipulation. These animals weren't born, they were assembled. While some see it as an advancement, others view it as an eerie overstep, tinkering with life not to heal, but to optimize. The dogs can't speak, but their bodies now carry the weight of choices made far above their understanding. Number 11. Genetically modified cyborg dragonfly. Inside a lab, a dragonfly was stripped of autonomy and rebuilt as a flying surveillance tool. Scientists implanted light-sensitive neurons into its spinal cord, wired them to remote systems, and fused tech to biology. A tiny backpack was strapped to its thorax, packed with sensors and microchips. The result wasn't just a bug, it was a controllable cyborg. This wasn't about nature anymore. It was a prototype for covert data collection, capable of flying undetected into spaces no drone could reach. Steering was handled by pulses of light, flashing directly into the dragonfly's brain and overriding its own instincts. The creature still flapped its wings, but not on its terms. It was piloted like a machine. These genetically engineered insects were sold as innovations for environmental monitoring and search and rescue work. But anyone paying attention saw the military potential. A living creature repurposed as hardware. The dragonfly had survived evolution for millions of years, until science decided it needed rewiring. Well, what we can say. Number 10. Genetically modified salmon. Well, this is insane. A biotechnology company quietly engineered a type of Atlantic salmon that grows twice as fast as its natural counterpart. To do this, scientists inserted a growth hormone gene from the Chinook salmon, fused with genetic material from an ocean pout, into farm-raised salmon eggs. The resulting fish could grow year-round and hit market size in nearly half the time. On paper, it was an agricultural breakthrough. In practice, it stirred serious environmental fears. What happens if these lab-altered salmon escape into the wild? Could they outcompete or even interbreed with native salmon species? The company promised safeguards, including land-based tanks and sterile fish, but nature rarely sticks to containment plans. Critics worried not just about the ecological consequences, but about a larger trend replacing natural food sources with genetically optimized alternatives. The modified salmon doesn't look monstrous. It doesn't grow fangs or turn violent. But its existence raises one quiet, unsettling question: what exactly are we willing to risk just to make food a little faster and cheaper? Well, everything. Number 9. Transgenic cotton and sterile insect warfare. In the cotton fields of Arizona, scientists declared war on a pest that had been ravaging crops for generations: the pink bollworm. This tiny insect had a reputation far greater than its size, chewing through harvests and wiping out months of effort. The solution wasn't pesticide. It was genetic sabotage. Researchers modified both the cotton plants and the worms. The cotton was engineered to produce a protein lethal to insects. At the same time, the worms were genetically altered to be sterile. The strategy was to flood the fields with these engineered pests and let nature run its course: no reproduction, no infestation. But this wasn't a simple plan. Farmers had to resist using traditional insecticides to allow the experiment to play out. It worked. The pink bollworm population collapsed, and the cotton was left standing. The method proved that genetic warfare wasn't just for science fiction. It was already here, woven into the soil, efficient. Yes. But what about the long-term ecological balance? Well, they're still hanging in the air. Number 8. Dolly the sheep. She wasn't the first cloned animal, but Dolly was the first mammal ever cloned from an adult cell, a breakthrough that rewrote the rules of biology. Born in 1996 in a quiet Scottish lab, Dolly came from a mammary gland cell taken from a 6-year-old sheep. Scientists fused that cell with an egg stripped of its nucleus, used an electric shock to trigger division, and implanted the embryo into a surrogate. Months later, Dolly arrived, genetically identical to the donor. Her birth stunned the world. Cloning was no longer theoretical. It had a face, a name, and a wool-covered body. Dolly lived a relatively normal life, grazing and aging like any sheep. But her existence set off a firestorm: ethics, identity, and the fear of what might come next. Was this the doorway to human cloning? Would the future be filled with copies instead of children? Dolly's body now rests in a museum in Edinburgh. Number 7. Monkey genetically engineered to have Parkinson's. A group of scientists in Japan created a monkey that developed Parkinson's disease on purpose. Not by chance, not by accident. They designed its brain to deteriorate. The logic behind it was cold but compelling. By inducing the disease in a living brain similar to ours, researchers could track how it spreads, where it begins, and how best to fight it. For medicine, this was a step forward. For ethics, it was a steep drop. The monkey showed tremors, muscle rigidity, cognitive decline, the same symptoms human patients face. It became a vessel for studying human suffering, mirrored in a silent body that couldn't consent. The hope is to develop treatments that could slow or reverse Parkinson's in people, but at what cost? The primate's brain was forced into a state of degeneration just so humans could learn how to delay their own. Even among scientists, the project stirred conflict. Animal testing is nothing new, but this. Number 6. The sudden death mosquito. It doesn't look threatening, but the mosquito is humanity's most lethal enemy, responsible for hundreds of thousands of deaths every year from malaria, dengue, and Zika. Scientists knew wiping them out entirely could save lives. But pesticides had limits, so they engineered a biological answer. Using genetic modification, researchers introduced a self-limiting gene into male mosquitoes. This gene causes their offspring to die before reaching adulthood. When released into the wild, these lab-born males breed with females, and the population collapses. It's a strategy that targets reproduction without chemicals. In places like Brazil and the Cayman Islands, the plan showed promise. In some areas, mosquito populations dropped by over 90%, but questions remain. What happens if the modified genes persist in unexpected ways? Could new mutations rise in response? Despite successful trials, the debate around genetically altering entire insect populations never truly settled. This wasn't a chemical war, it was genetic disarmament. Number 5. The Vacanti mouse experiment. Back in the 1990s, a photo began circulating that unsettled just about everyone. A hairless lab mouse with what looked unmistakably like a human ear growing out of its back. The image was real. Scientists Joseph and Charles Vacanti, along with MIT engineer Robert Langer, were testing a new form of tissue engineering. The ear was made of biodegradable scaffolding shaped like a human ear and seeded with cow cartilage cells, designed to explore the future of lab-grown human parts. The mouse was a test host, not genetically modified, but still caught in the middle of science and spectacle. Their goal wasn't horror, it was reconstructive medicine. Ears are notoriously difficult to rebuild surgically, and this was supposed to be a solution. But the image struck a chord. Something about seeing a human feature on another creature triggered unease. The research itself didn't go far beyond the prototype stage, but the mouse became a symbol. Not of success or failure, just a chilling reminder of how weird progress can look in its earliest form. Number 4. See-through frogs. Well, we know we've talked a lot about this thing. Japanese researchers at Hiroshima University developed a frog whose skin was selectively bred to be semi-transparent, not for show but to eliminate the need for dissection. With visible internal organs, blood flow, and tumors, scientists could monitor biological processes in real time while the animal stayed alive. On the surface, it sounds humane, a way to reduce the number of animals sacrificed in research. But what it created was a living, breathing window into experimentation. These frogs can't hide anything. Not stress, not illness, not the effects of chemical injections or radiation. They're constantly observed, their bodies transparent, but their suffering less so. While this advancement helped in areas like cancer research and drug testing, the frogs became trapped in a paradox, alive for longer, yet still subjected to invasive tests. They may have helped delay death in other creatures but couldn't escape its slow arrival themselves. It's a form of mercy wrapped in scientific coldness. You can see their hearts beat, but that doesn't mean they're at peace. Number 3. Ghost hearts grown in bioreactors. At the Texas Heart Institute, scientists built something that looks like it belongs in a dystopian future: hearts stripped of color, bloodless, suspended in bioreactors, waiting for new life. These are ghost hearts, cadaver organs washed of their original cells, leaving only the collagen scaffold. Into this hollow frame, researchers inject a patient's stem cells. Artificial lungs and pumps simulate the body. Slowly, the heart begins to rebuild itself. The concept is revolutionary. Transplants without rejection. No need for lifelong immune suppression, no waiting lists, but it's also haunting. These hearts float in sterile chambers, surrounded by machinery. They have no pulse, until given one. Doris Taylor and her team have engineered over 100 of them so far. Each iteration is a step closer to lab-built organs tailored to each patient's DNA. It's medicine's holy grail, but with an eerie aesthetic: organs without origin, flesh without history. It's easy to marvel at the science, harder to ignore the feeling that somewhere along the line, something intimate became industrial. Number 2. Belgian blue. From a distance, it looks like a cow that's been photoshopped. Up close, it's even stranger. These cattle carry a rare mutation known as double muscling, which prevents the body from producing a protein that limits muscle growth. The result is extreme muscle mass, meat stacked on meat, engineered not in the modern CRISPR sense, but through decades of selective breeding intensified by artificial insemination. The breed was fine-tuned in the 1950s by Belgian scientist Professor Hanset, aiming for leaner meat and rapid growth. What he achieved was a cow that looked like it belonged in a comic book. But the mutation comes at a price. EAVs are often born via cesarean section due to their size. Many suffer from joint issues, reproductive complications, and shortened lifespans. While beef is praised for being low-fat, the ethics behind breeding animals for visual mass, with no regard for function or comfort, cast a long shadow. Number 1. Human-pig hybrid, created in the lab. Researchers in California once created a chimera embryo combining human stem cells with a pig embryo, a biological blend that stirred more controversy than it did celebration. The goal of the experiment was to find a way to grow transplantable human organs inside animals. If successful, it could solve chronic donor shortages. But mixing human DNA with another species opened a minefield of ethical and biological risks. There were real concerns that human cells could migrate to the brain, raising the horrifying possibility of animals developing partial human cognition. The embryo was destroyed after just a few weeks, but the damage was already done. Public backlash questioned whether science had crossed into dangerous territory. Scientists insisted the research was carefully contained, but it didn't ease the unease. This wasn't fiction, it was a real attempt to farm parts of humans inside livestock. And while the headline faded, the experiment left behind a trail of questions no one has yet been able to answer cleanly or ethically. If you've enjoyed this video, please don't forget to like, share, and subscribe for more interesting content like this. Thanks for watching, and we'll see you soon in the next one.